Hi, this is Duncan Ferguson. In this unit, we're going to talk about the molecular mechanisms of thyroid hormone, focusing on, of course, the genomic effects or nuclear receptor effects, but also others directed at the mitochondrium and the plasma membrane. So in this unit, we're going to quickly review the structure of thyroid hormones and then move on to talk about the nuclear receptor, signal transduction mechanisms, a little bit about how nuclear receptors work, the structure of the thyroid hormone receptors, various subtypes, then the action of thyroid hormone on mitochondrial receptors, particularly those that impact calorigenesis and thermoregulation. And then finally, a relatively new area of understanding, the integrin receptors that seem to be detecting thyroid hormone and the actions associated with those which do not require a genomic mechanism. So thyroid hormones are iodinated compounds. They are basically linked tyrosines and are small molecular weight compounds that range from a molecular weight of 651 for T3 to 777 for T4. So they're relatively small lipophilic molecules. When we talk about the active thyroid hormones, we're going to talk about basically T4, and we'll see that it has lower general metabolic activity than T3, but T4 does have some of its intrinsic activity. And then there's reverse T3, which basically is devoid of most general metabolic effects. There are some other mechanisms being discovered for reverse T3, but we won't go into those in this unit. So in this graphic, we show the thyroid hormone receptor, which acts as a heterodimer with the retinoic acid receptor to interact with DNA. You can see here, heterodimer, we've got retinoic acid on the left interacting with its receptor, and then T3 as a prototypical most active thyroid hormone on the right, and these work together in a dimer that of two different receptors to then impact by binding on the hormone response element, or the thyroid response element is another term for it, leading to alteration of genetic transcription, messenger RNA, and protein synthesis. So it's interesting to compare the receptor affinity, the nuclear receptor affinity, or binding tightness for these analogs of thyroid hormone on the left with their biological or metabolic activity. And so you can see that the highest amount of activity, we're setting it to 1, is LT3 levo triiodothyronine. The D form has activity, but it's less, and you can see so does the biological activity go down. Then we have T4, let's focus on that first, and you can see it's about, and depending on the study, somewhere between 1/10 and maybe 1/3 of the activity of T3 in these systems, whereas reverse T3 is essentially devoid of activity, both with nuclear receptor binding and metabolic activity. The product, the metabolite of T3 called triac, which is a deaminated product of triiodothyronine, you can notice here that it has a high nuclear receptor binding and has some metabolic activity. So this is a little bit in discordance. But I mentioned that product because it's being used inappropriately and unwisely, I guess I should say, by some people trying to lose weight because it's something that they can take and help them lose weight, but it leads to, as we point out, many side effects such as heart arrhythmias. In this slide, I just want to have you hone in on the take-home message, but for those of you interested in how different subtypes of the thyroid hormone receptors are located in different places, I think it's very interesting and useful, and you can read that. But the main thing is that with multiple forms of the hormone receptor, there are both tissue differences as well as developmental stage differences in expression that help control sort of the vast diversity of complexity of the physiological effects of thyroid hormone. Here I want to basically show some basic concepts of nuclear receptor signaling, and on the left we show how a binding of in the ligand binding domain, that's what LBD is, in the DNA binding domain, and then the hormone response element of a nuclear receptor mediated hormone. One effect can be to attract a co-repressor to that complex, and it leads to repression or reduction of expression of a target gene. On the right-hand side, you have just the opposite. You can have a scenario where, for whatever reason, that particular ligand binds to the receptor and the DNA binding domain, the hormone response element, and attracts a co-activator to lead to activation of messenger RNA. So we can have these two general effects on either upregulating a messenger RNA or downregulating it, depending on the way things are programmed. So returning to the thyroid hormone, remember we said it's a heterodimer with retinoic acid. The heterodimer complex affords the greatest affinity binding and is thought to be the major functional form of the receptor. And this is in contrast to steroid hormone receptors. Thyroid hormone receptors basically can bind the DNA in the absence of the hormone. So this leads to repression of the activity of whatever gene is associated. So that means, if you go back to the previous slide, it's attracting a co-repressor. And that co-repressor complex has what's called histone deacetylase activity, which by altering the conformation of chromatin can basically turn it off. Once you get hormone binding to this complex of the heterodimer, in this case the thyroid hormone receptor, you have a conformational change that leads to attraction of the transcriptional activator, and that co-activator, and that sort of turns that gene on again. Let's now take a look at the action of thyroid hormone at the mitochondria, and I think it's useful to look at it from an integrated standpoint first. This graphic shows, and it's mainly focusing on the proteins that are synthesized through nuclear receptor transcription at the nuclear complex. And in this graphic, I want you to note that thyroid hormone is involved with synthesizing enzymes that make lipids, enzymes that are associated with metabolism of lipids in the liver, therefore generating fatty acids. It's involved with the synthesis of carnitine palmitoyltransferase, which is a major transporter of fatty acids into the mitochondrion. That's what this is here, mitochondrion. And of course, it stimulates many of the complexes, we'll go into that in the next slide, that are associated with oxidizing or utilizing energy stores such as fatty acids and turning them into basically energy. So thyroid hormone basically is this orchestra director of all of these things that impact both the availability of substrate as well as its oxidative metabolism. A little more on mitochondrial action of thyroid hormone. We believe that some of the more rapid clinical effects of thyroid hormone are probably due to interaction directly with receptors on the inner mitochondrial membrane. These can lead to very rapid response of oxidative metabolism. There are also the metabolite 3,5-T2 very recently has been determined to have similar effects at this receptor. More delayed effects of thyroid hormone on mitochondrial respiration probably do relate to this, what I showed you in the previous slide, insofar as that it requires nuclear receptor activation. These include changes in phospholipid turnover, stimulation of uncoupling proteins, we'll talk about that in the next slide, which are important for uncoupling the hydrogen ion gradient and therefore making sort of inefficiency to the oxidative phosphorylation and therefore leading to non-shivering thermogenesis. So both uncoupling proteins and this nucleotide translocase increases inner mitochondrial membrane hydrogen leak and increased non-shivering thermogenesis. And thyroid hormone is absolutely crucial to the generation of heat in forms of non-shivering thermogenesis. This is why, for example, a very young animal like a puppy or a kitten that is, once it's born, cannot thermoregulate, needs to stay with its mother, and this is at a time when usually their eyes are not open. And that is also thyroid hormone dependent. Thyroid hormone will increase, open their eyes. It will lead to the increase in their ability to generate enough heat to be independent from that standpoint from the mother. A little review of the biochemistry is probably in order here, how heat is generated by the effects of thyroid hormone. So if you look at the process of oxidative phosphorylation in the electron transport chain in the mitochondrial membrane, which is shown here, you have the generation through all this process of hydrogen ion gradients. And so when this hydrogen ion gradient is coupled through the process, you get ATP generation. And of course, the ATP is exported by the adenine nucleotide translocase here outside to allow cellular work. So that's the coupled reaction. And then there's the reaction that's associated with an uncoupled situation where the hydrogen ions basically are allowed to leak through, and this energy that's been generated is wasted, if you will, and forms the non-shivering thermogenesis through that inefficiency. And as we mentioned, basically, thyroid hormone can increase the not only the uncoupling proteins, so thyroid hormone has an action there, it can also increase the formation of these nucleotide translocases that have an impact on efficiency of the whole process as well. And finally, a relatively new and an exciting area of thyroid hormone action has been the recognition that thyroid hormone or metabolites of thyroid hormone can act at the plasma membrane without necessarily involving nuclear action. And so in this graphic, what we show here is that integrin receptors right here are interacted with by T4 and T3, and they then have crosstalk with the epidermal growth factor and fibroblast growth factor receptors, but ultimately act through ERK1 and 2, which are kinases, and serine phosphorylation leads to result. It does interact with the nucleus with these transcription factors that alters at the end of the day a messenger RNA and the secretion of proliferative and pro-angiogenic proteins like fibroblast growth factor and epidermal growth factor, and they interact with their receptors. So why is this important? Well, this particular pathway has been explored recently to be blocked by a metabolite of thyroid hormone called tetrac, which is a deaminated T4 version, and that tetrac can reduce this integrin type of action of thyroid hormone, and therefore it's been explored as an adjunct to reducing the angiogenesis that occurs in cancer, for example. And so this is a very exciting new area in action of thyroid hormone. So in summary, T3 and with lower affinity T4 bind the nuclear thyroid receptors that mediate what we should call the medium or long-term effects of thyroid hormone by increasing or decreasing messenger RNA and protein synthetic rates. And when we talk about integrated effects of thyroid hormone, we'll be looking at things like, loss of hair, altered lipid metabolism, etc. There are 4 forms of the thyroid hormone receptors, the knowledge of which specific ones are probably not important to you right now, but the fact that there are those and that they're heterodimers that interact with the retinoic acid receptor is an important thing. The thyroid hormone receptors have tissue and stage-dependent differences in expression, leading to the complexity of the response of physiological effects of thyroid hormone. The mitochondrial effects, thyroid hormone via the adenine nucleotide translocase and uncoupling proteins have been described, as well as nuclear-mediated effects that increase uncoupling and non-shivering thermogenesis. And finally, the integrin plasma membrane receptors stimulate transcription factors that enhance proliferative and pro-angiogenic growth factors. And so these, because they seem to stimulate angiogenesis, these have been explored as agents to try to reduce cancer metastasis. And so that's an exciting new area.